Okay, what we're going to focus in on today is the fundamentals of microbial growth. What you're going to find is that this relates not only to how microbes grow, but how we can stop or inhibit their growth, and we'll move forward. Now, microbes show dynamic and complex growth in nature and more distinct growth stages in the laboratory. Microbial growth is defined as cell division that produces new daughter cells and increases the total cell population. This is extremely important in healthcare issues such as biofilms. It helps us with understanding the variables of microbial growth, helps us to comprehend the challenges of biofilm growth, control, and eradication. If you take a look here, this is a biofilm that's grown up on a catheter. The organism is alkagenous, and it's a gram-negative bacteria. So what you have to keep in mind is that biofilms act as protection for the bacteria against immune processes like antibodies, T cells, macrophages, but they also protect against chemical assaults like antibiotics. Now, bacteria usually divide by binary fission, but some may use budding or spore formation. Budding is an asexual means of reproduction, similar to binary fission, but the bud is smaller than the mother cell despite the equal chromosome copy present. Spore formation, for example, in fungi, can be sexual or asexual, but in bacteria, the process is asexual. Not to be confused with formation of endospores. Spores are cells with special formations that are an exact copy of the bacterial cell. When we talk about generation time, this is the time required for specific cell type to divide. Let me give you a simple explanation of this by using E. coli. The generation time for E. coli is approximately 20 minutes. What does that mean? It means if you start at the top of the hour with one cell, at 20 minutes past, you're going to have two cells. At 40 minutes past, you're going to have four cells. And when you complete one full hour, you're going to have 8 cells. Now, if you take that doubling, okay, and basically you take a 24-hour period starting with just one cell. In a 24-hour period, because you're going to have about 72 divisions, you would have the total amount of cells that basically equates to 2 to the 72nd power minus 1, well over a billion cells. Now, exponential growth. This is a growth rate that becomes more rapid in proportion to the growing number or size. Here we have an example of several different cells. This is more sporulation here. This is binary fission. You see the cells equally separating out. Here you do not. Now, as I bring this back to you with this nice chart here, you can see that by the time you get done in 1 hour, you've already hit your third generation. And there are a lot of cells. Now, this does not mean that you're going to see the same time period for generation time for other types of bacteria. Some of them may differ. For example, tuberculosis bacteria, because of the waxy coat that exists in their cell membrane, mycolic acid, they're going to have a much slower rate of reproduction. Okay, roughly about 18 to 24 hours for a generation time. Now, bacteria have 4 distinct growth phases when they are cultured using a closed pure batch system. Keep in mind when I say a closed pure batch system, this has a set amount of nutrients, it has a set amount of oxygenation, pH control, temperature, etc. When you take your first cells and you add them, you're going to have a lag phase. This is a delay phase that occurs when bacteria cells are just adjusting to the new environment. Then you have the log, or otherwise known as logarithmic phase. This is a phase of rapid Exponential growth. It's demonstrated by an upward sloped line plotted by a logarithmic scale as a function of time. Then you get a plateau. That's the stationary phase. The plateau phase, where the number of cells dividing equals the number of cells dying. Next, you have the death phase. Now, you have to keep in mind something about death phases. This is due to the buildup of waste and the depletion of nutrients. This is the rate of cell death will exceed the rate of cell division. It's also referred to as a negative slope line. One thing you got to keep in mind when you go into the death phase, if you hit that, you really can't be assured the type of cell that you started off with is going to be exactly the same once you hit the death phase, because cells may have some mutations. So you will now have those that thrive a little bit better in lower amounts of nutrients. They may actually use some of the waste as a nutrient. They may feed on other cells and things like that. So they're going to be somewhat shifting toward those cells that can handle a more sparse, austere environment. It may not be representative of what you originally started for the cell culture. Death phases are usually something where it's like, well, unless I want to study bizarre biological behavior, if I'm making this a cell culture for, let's say, growing something that might produce a biological drug or manufactured agent, you pretty much have to dump the whole batch now. Now, a chemostat, this is a closed batch system for culturing cells and cell products. This is where the rate of nutrients is continuously added and adjustments to the pH match the rate of removal of excess media, excess cells, and media or waste. To give you an idea of this, if I have a closed system here, I'm going to be slowly taking out some in these chemostats. But let's just show you in this chart here. First, we had the lag phase, cells adjust to their new environment. Then we have this ramping up, the log phase, exponential cell growth. Stationary phase here, number of cells dividing equals number of cells dying. No real changes in the number of viable cells, but then you start going down the death phase. You have a buildup of waste and dead cells. Cells die as waste accumulates and nutrients are depleted. Okay. Here is an example of a good chemostat. Now, one of the key points here is this. You're always going to have air coming in. You're going to have air coming out. You're going to have a feeder tube that delivers fresh nutrients. And of course, you're mixing everything. You're also going to have a constant amount of heat. You're going to have a pH monitor tube in here. And you're also going to, as you add, let's say, I'm just going to pull a number off the top of my head. Let's say 5 mils every minute or so. I'm going to pull 5 mils of liquid out. That's going to basically remove some of the waste, some of the cells, etc. So you keep this hopefully at what you would expect to be somewhere in the range back at the stationary phase. Now, if you notice, there's lots and lots of tubes here. These tubes aren't always delivering something. They may be pH monitors, they may be temperature monitors, they may be oxygen sensors, etc., to make sure that the conditions are favorable for continuing a nice balance of cells that you have present here. Now, prokaryotes can adapt to various growth conditions. When we talk about temperature, there's a maximum and minimum temperature, the upper and lower temperature ranges that support microbial growth. There are the concept of the optimal temperature. This is the temperature where the growth rate is highest. So certain organisms will thrive at an optimal temperature. But when we say optimal temperature, well, that depends. Here's the key. Psychrophiles are microbes that thrive at minus 20C to 10C. Psychrotrophs are microbes that thrive at 0 Celsius to 30 Celsius. This is where you have many foodborne illnesses are caused by these particular types of organisms. Mesophiles are microbes that thrive in the temperature range of 10 Celsius to 50 Celsius. Thermophiles, these are microbes that thrive at 40°C to 75°C. So what I'm trying to say here is that this class here, let's say psychrophiles, their optimum temperature is somewhere in the range between -20°C and +10°C. Now, there are some also extreme thermophiles. These microbes thrive at temperatures at 65 to 120°C. Now, if you get a little confused there, we're talking at 120°C, that's well over the boiling point of water. Well, how do they live? It's usually underwater, under high pressure. The pressure is such that it does not allow water at a temperature of 212 Fahrenheit or 100 degrees Celsius to boil. But it's nice and warm and proper and toasty for thermophiles. Now, not only do we have temperatures that are optimum, but we have optimum pH conditions. Okay? Microbes have a favoring of a pH. Some of them are neutral, so they would be neutrophiles. Microbes that grow at a pH range of 5 to 8. But acidophiles, these are microbes that grow in a pH from 1 to 5. For example, some of them may exist in hot springs and volcanic vents. Alkalophiles, these are microbes that grow in the basic range of a pH from 9 to 11. Then we have those that thrive in high pressure. These are called barophiles. You'll find them such as deep under the ocean. There are some also that have a favoritism, some that favor an environment that is extremely salty. Okay? These are called halophiles. These are microbes that thrive in high salinity. Example, 35% salinity. Now, usually when you start getting these really extremes, this is where you get into the class of extremophiles. Most of them are archaea, but they are organisms that thrive in the very extremes of pH, salinity, temperatures. And these are some organisms that in many cases are believed to have been around well before Earth really was an oxygen favorable environment. They still exist in areas of high salinity, extreme pH, and or extreme temperature. And you can see this. As you can see, psychrophiles, they would be down here in probably some frozen lakes and things like that, or in Arctic or Antarctic environments. When we get to the human body temperature, that's roughly 37 degrees Celsius, that's where you're going to find the existence of mesophiles. Remember that psychrotrophs are the ones that would be in the range of, let's say, refrigeration, etc. A little bit above that, a lot of these are foodborne illness type of organisms. Okay, enough of that. Let's move forward. By the way, this is a highly salty area. And yet, if we looked for microorganisms there, we'd find them. These are actually all the hardened salts that you would see as the water level dipped down. Now, microbial growth. You have to be mindful of the fact that you're going to have organisms that when they go through binary fission, they'll have a space between the cell wall and the plasma membrane. Sometimes this is an important shield against certain extremes in chemistry, osmolarity, things like that. Let's move into oxygen requirements, because this is very important that you get this down now, because it relates to how some organisms thrive in environments with lots of oxygen versus environments that have no oxygen. You have to introduce you first to the concept of reactive oxygen species. These are referred to as ROS. These are molecules that contain oxygen, but are highly reactive to other substances. So they could be a single oxygen atom, and so they would have a negative charge, O, or O minus 2. Some organisms require oxygen. They've developed the means to detoxify ROS. When we put the term obligate in front of it, obligate aerobes. That's required. They have to have that oxygen. These microbes have an absolute dependence on oxygen for cellular metabolic processes. If I talk about something that is a microaerophile, these are microbes. They require small amounts of oxygen and they limit their ROS exposure. Now, anaerobes, whenever you put AN in front of something, it's sort of not. So, not aerobes. Anaerobes are organisms that do not use oxygen for their cellular metabolic needs. Something that's aerotolerant for anaerobes. These are microbes that tolerate the presence of oxygen but do not use it for their cellular metabolic needs. Obligate anaerobes. These are microbes that cannot use oxygen and cannot survive in the presence of oxygen, as they cannot eliminate ROS. Now, when we talk about using the term facultative, this means helpful, but usually helpful but not necessary. So when I say something is a facultative anaerobe, these are microbes that have enzymes to detoxify ROS, and they switch their metabolism to use oxygen or employ fermentation as an anaerobic form of metabolism. You want to see some of the pathogens and oxygen profiles on the body, you will review table 7.1, which would be right here. For example, let's start off with the skin. You have facultative anaerobes there, the Staphylococcus aureus type. Proprinnium bacterium acinase is an aerotolerant anaerobe. In the stomach, you have the microaerophile known as Helicobacter pylori, which is responsible for ulcers. In the large intestine, now think about this, there's not lots of oxygen there. So you have Clostridium difficile, which is an obligate anaerobe, and then you have the Salmonella species, which is more a facultative anaerobe. With lungs, though, Bordetella pertussis, whooping cough, is an obligate aerobe. The Mycobacterium tuberculosis is a facultative anaerobe, whereas Mycoplasma pneumoniae, that is an obligate aerobe. It's got to have oxygen. The blood and lymph, you will find Bordetella burgdorferi, the agent that causes Lyme disease. This is a microaerophile, whereas Treponema pallidum, which causes syphilis, is a microaerophile. And Yersinia pestis, the agent that causes plague, is a facultative anaerobe. Moving forward, here's an example that you might see as oxygen concentration goes from high to low. If you were to culture these in tubes, this is what you might find for where the colonies set themselves up. Obligate aerobes are more closer to the top of the tube because obviously you're going to have gas coming up underneath the cap down into the tube. Obligate anaerobes, they're going to be put down at the bottom. Now this is if I believe if we reuse this for broth as opposed to gel, the agrigel. Now microaerophiles might be below the immediate surface, but within some range to get a little bit of whatever is necessary for their oxygen. Whereas facultative anaerobes, hey, they can be at the top or they can be clustered throughout up all the way down the bottom. Aerotolerant anaerobes can be scattered throughout the entire tube. Now, microbes require nutrients, they require growth factors, and a source of energy. When we talk about essential nutrients, these are required nutrients for microbial growth and can be organic or inorganic in the microbe's environment. To help you to understand this, keep in mind, when I say organic, it's something that is attached with carbon. Inorganic, something that is not connected with any carbon atom. So heterotrophs are microbes that require external sources of organic carbon. The organic carbon might be sugar, lipids, proteins. So these are organisms that may have to live off cell membranes or get them from basically infecting cells or infecting the extracellular matrix, the fluid around the cells, etc. Autotrophs are self-feeding. These are microbes that perform carbon fixation using some energy source, such as sunlight. So these might be those organisms that use the sunlight to make the carbon fixation to make their sugars, lipids, proteins, etc. Carbon fixation is a process that requires energy to fix inorganic carbon, such as carbon dioxide, into organic carbon compounds. Now, let's move from carbon fixation to nitrogen fixation. Okay, remember, nitrogen is key for amino acids. Microbes that take nitrogen from the atmosphere and convert it to nitrogen compounds for cellular uses, such as amino acids, ammonia, that's nitrogen fixation. When we talk about growth factors, these are compounds that microbes need to sustain growth. When we talk about something that is a fastidious microbe, these are microbes that need multiple growth factors for culture. Some microbes have evolved to depend on host organisms for many of the growth factors, lipids, amino acids, etc. For example, some organisms are very difficult to culture because they are, in fact, fastidious. And yet, if we use a culture medium and it includes some unique materials such as, sheep blood, might include human plasma or fetal bovine serum, something like that, it contains a variety of unique compounds that act as these specific growth factors and allow for the culturing of the microbes. Now, energy sources. Well, we got to start talking about where do we get the energy that require energy to grow. All cells do that. If I talk about a phototroph, these are organisms that use light for energy. If I talk about a chemotroph, these are organisms that break down chemical compounds for energy. That's kind of like what you and I are. We have, uh, we consume food, we break it down to basically the chemical compounds, we convert these chemical compounds into energy. Our energy would be in the form of ATP. Now, this is where you're going to break down a couple of different levels. First, you got to find the energy source. Then you got to find the carbon source. So if I talk about phototrophs, I can have photoautotrophs. Auto should give you the clue right off the bat. Self. Photoautotrophs have an inorganic carbon source, usually carbon dioxide. They use sunlight, such as cyanobacteria found in freshwater environments. The energy from the sunlight provides them with the necessary means to make carbon compounds. Photoheterotroph. The carbon source is organic, so it might be methane, it might be even sugars, etc. You have Helobacterius mobilis, which is found in rice paddy fields. So it still uses an energy source for some of its processes, but it's still dependent on organic compounds for its carbon source. Now move over to the side where we get into chemotrophs. Nutrient breakdown, chemoautotroph. Its organic, its carbon source is inorganic, usually again carbon dioxide. Thiobacillus denitrificans is found in soil, mud, freshwater, marine sediments. So it uses certain chemicals to break down, provide the energy so that it can take inorganic compounds and make them into organic compounds. Now, chemoheterotroph, hetero should tell you right off the bat, other sources. And so with Escherichia coli, it's going to get organic carbon sources, glucose, for example, things like that. It's a common inhabitant in mammalian intestines. Now, microbes are grown using various media. Now, this is where we get into how we use the media, how we use it and culture it, and all these other wonderful things. The physical state of the media is important. We have liquid, solid, and semi-solid. All of this is dependent upon agar. It was easy to make liquid media. We called it broth media. The problem was sometimes you wanted to see what colonies were being formed. The problem you could not use was gelatin. because basically the the materials that make up gelatin could be broken down by the bacteria. So another suggestion came up: agar. Now this is a powdered polysaccharide for solid and semi-solid formats. It's extracted from seaweed. Now with broth media, you have simple microbial media with the select components, and of course you're going to add sterilized water. You want to prevent any contamination. Now when you grow these, you may grow them on a petri dish, you may grow them on a slant or a deep. What's a slant? That's where you're going to have solid media. It's added to the tube, and then it's allowed to cool, but it's at a slanted angle. So what happens is, instead of a short little circle of growth area at the top, you'll have a much more elongated angle, sort of like an oval. That allows for greater growth surface area. An example of this would be the Simmons citrate test and the triple sugar iron media test. Now a deep, this is a little bit different. Again, you're using solid media. You can put them in test tubes, but the test tubes are placed upright, not at an angle, because what you're going to do is you're going to have a reduced amount of agar, and you're going to have it just solid enough so it holds together. And you're going to take a fine needle and you're going to stick the microbe into the, let's say, the middle of that solid media. A deep is used as a microbe motility test. In other words, how microbes will move. Seems to work out. And you can see here these test tubes here with a variety of different medias. Now notice that some of them are turned upside down. In other words, what you've done is you've inverted the petri dish. Why? Because if you incubate it, you don't have the chance of having droplets of liquid, or in other words, evaporation, condensing on the top of the plate. You don't want that because it makes it harder to see and read the plate. You also don't want it because it may change some of the actual chemical composition or conditions in the agar. But you see all these test tubes here, how they have a nice round surface? Okay, so these might be broth media, for example. Now, we get into the media. We have defined and complex. Defined, think about this. If I ask you, what's the definition of this? What's the makeup of a bottle of Snapple? What's the makeup of Coca-Cola? Well, that's top secret according to Coca-Cola fans and Coca-Cola Corporation. But defined, you could also say a synthetic media. Basically, what you've done is you've chemically been able to develop a precise, exact media. It's composed of so many grams of this, so many micrograms of that, so much of this, so much of that. You exactly know the organic and inorganic compounds. You want to be noting that additional growth factors such as amino acids or vitamins may also be added. Especially when you're dealing with some organisms that cannot synthesize a particular amino acid, you have to add that to the media just so that the organism can thrive on that media. Now, complex media is different. It's enriched, contains a mixture of organic and inorganic nutrients that are not fully defined. This is where I get back to what I was talking about earlier. It may contain a complex series of ingredients like blood, milk proteins, yeast extract. This is usually for fastidious organisms with complex growth requirements. The milk proteins, the blood, calf serum, any of these things, yeast extract, will contain a variety of small amounts of various different types of growth factors that make it excellent to grow to grow the organism that you're looking for. Now, the other thing that you were going to go through is differential media and selective media. You may have dealt with this already in your lab exercises, but differential media is a specialized media formulated so they can identify the organism And so the identity of it can be visualized based on how the organism metabolizes media components. Maybe it'll cause a color change. Maybe it will, the colonies themselves will change color. Usually, though, it's causing some sort of alteration in the media. It may change color, it may turn in some unusual means, but it's very clear what you're going to see. Selective media singles out certain microbes that have specific properties. It includes ingredients in the media that foster the growth of one organism, but suppresses the growth of other organisms. It may have, for example, biosalts in it that certain organisms would not be favored, but other organisms would have no problem with. Here was an example that we saw the Simmons citrate test, and here's the mobility test. So this would be a slant. This would be a deep. They took the needle with the organisms that were on that needle and you stab it down deep. Okay? Here is our complex versus defined media examples. Now, with Luria Britannia media, and no, you're not going to get tested on how many grams of this or that. Don't worry about that, but get the idea behind it. You're going to mix this in 1 liter of liquid media, so obviously you're going to have sterilized water. Okay? You're going to have a couple of basic compounds, but then you're going to add other components, sugars, amino acids. These will be provided by complex yeast or protein preparations. You don't know the exact level, but if you bust open yeast cells, they're going to have a variety of amino acids and sugars and other components. If you take soy protein, take from the soybean, you take a certain amount of protein, add it to that, it's going to favor certain things. All right? That's complex. Now, defined is you're just basically, you can go down to the chemistry department, you pull out the bottles of glucose, magnesium sulfate, calcium chloride, disodium phosphate, potassium phosphate, sodium chloride, ammonium chloride. Yeah, add it to water, measure these amounts out, there's your medium. It's called glucose minimal salts medium. Okay? You don't add the vitamins and amino acids to these minimal slats media. If the organism to be able to grow, if it needs some nitrogen, you may add ammonium chloride and let the organism use the ammonium chloride, the ammonium ions, to basically build all the necessary amino acids. All right. Now, let's go over a couple of examples of both differential and selective media. If we use blood agar, the additive of sheep red blood cells to the agar, the red blood cells are used as a nutrient and as a differentiating indicator. For example, certain bacteria will undergo either alpha hemolytic, beta hemolytic, or gamma hemolytic changes. Let's take a look at each one of them. Alpha hemolytic, the bacteria turn the blood green, and this is due to oxidizing the hemoglobin. Beta hemolytic, the bacteria make hemolysin, and they lyse the red blood cells and turn the media yellow. Gamma hemolytic, the bacteria do absolutely nothing to the red blood cells, and there's no color change. Okay. With selective media, though, they single out bacteria that have specific properties. For example, mannitol salt agar, MSA. This is selective due to the high salt content. So basically, microbes that ferment mannitol, which is a sugar, or it's actually a sugar alcohol, turn the media bright yellow. If there are non-fermenters, no color change. Now, the red comes from a pH indicator so that you understand that. Okay, so if you start having some pH changes, the pH indicator will tell you exactly what's going on. Here's one, eosin methylene blue agar. It has selective and differential capabilities. It contains the dyes eosin and methylene blue. These will limit the growth of gram-positive bacteria growth, while at the same time it allows gram-negative bacteria to grow because it has lactose. Most lactose fermenters appear as dark pink colonies. But let's say you had E. coli or some other bacteria with a strong acid production during the lactose fermentation. This is going to appear with a metallic green colored colonies. Isn't that cool? Wow. Now, gram-negative bacteria that do not ferment lactose, such as Pseudomonas aeruginosa or Salmonella enterica, will appear colorless on EMB agar. Here, we had the sheep blood, and you've got alpha here, that greenish coloration, beta here, that yellowish, and gamma hemolysis where there's no change at all. When we had mannitol salt agar, here we have mannitol that's not fermented. No, well, it's kind of turning a little, it still stays red, okay? But mannitol fermented, look at that. You got a definite color change all around it. And by the way, if we use eosin methylene blue agar, look at this. Lactose non-fermented, no major change. Lactose fermented, strong acid main, look at that green metallic color. And then the lactose fermentation, you can see the color there. So overall, these are ways to find out the identity of actually some of these organisms by using specialized media. Keep in mind that just sitting there and identifying a cell as, well, it's round. Okay, that's fine. There's a bunch of different species that are round. Well, it's rod shaped. Yeah, that's fine. What else does it do? The identity of microbes is partly accomplished by using selective medias that will favor or provide clues to its biochemistry. Okay, hope that helps. Now let's talk about anaerobic media because that's media that prevents microbes in a sample from being exposed to oxygen. How do you do that? Oxygen exposure will kill anaerobes. Now, anaerobes make up about 38% of the bacteria population in a wound. That's serious. Usually what you do is you add a compound that converts molecular oxygen to water, and that's called thiogluconate. You also have the anaerobic jar. This is a chamber to culture anaerobes, in which the culture plate will have an oxygen scavenging packet added to purge it from the culture media air. So basically what you do is you take your large cylinder chamber, add your culture plates, add this oxygen scavenging packet, close it up, and basically what it does is it reacts with the oxygen to sequester the oxygen, and what you have left is a nitrogen and somewhat carbon dioxide environment. Fine. But let's say you're doing a lot of this work. That's where you get into an anaerobic chamber. It's airtight, it's a controlled atmosphere chamber. It allows for manipulation and culturing of many samples of anaerobes at the same time. Now look, here's this smaller chamber, and here is that oxygen sequestering packet. Okay, then I can put that right in that whole assembly here into an incubator. But let's say I'm doing a lot of them. That's where you have the glove box. You see how that's specialized seals, and you will even have a monitoring of the amount of oxygen or the amount of carbon dioxide they have in there. Oxygen would be obviously very, very low, if not nothing. Here is a sealed airport. I could have said an airlock, I should say. And really what they would do is they would purge out the air. They would take a sample, either move it out or move it in. Before you move it in, you basically purge out all the oxygen, etc. Now, the next thing we're going to be going into, as you can see here, is how do we set up colonies on a culture plate? Okay, notice the first time you go back and forth, back and forth, back and forth. that's laying on a smear, but you know what? You see what happens after a while? You let it culture. Ugh, it's billions of cells right here. So the next thing you do is you turn it 90 degrees and you again sweep it back and forth for a little bit. Turn it again 90 degrees, sweep it again back and forth, back and forth, back and forth. By the time you should get to the third, if necessary, the fourth sweeping back and forth with this loop, you should start getting individual cells smeared out, which that individual cell will then grow into a full colony. Okay, moving forward. This is how we talk about collecting, isolating, counting, and identifying microbes. Because, hey, it's one thing to have 8 billion cells in a small sample test tube. You've got to get them to individual cells to really study them. Now, aseptic techniques are extremely important here. Aseptic techniques are methods that prevent the introduction of contaminating microbes to a patient, to a culture sample, or to a healthcare setting. Collecting samples in a clinical microbial analysis is extremely important in keeping in mind your aseptic techniques as you're doing this. For example, the streak plate technique, which I showed you just a minute ago, that's used to isolate microbes. The colony that you see, the little round ones, should appear to start off from one cell. That allows you for counting microbes. You can also use that to have one reliable colony that you can then set up a complete separate culture on. Okay, you'll know that it's pure and you'll know that it's separate. Counting cells is based on a small portion of the sample. There are cell counting chambers. These are called hemocytometers, and they're an instrument for visual counting of the number of cells, for example, in a blood sample or other fluid under a microscope. Now, there are other direct methods of measurement. Flow cytometers. These are cell counters that use fluorescently labeled cells, which pass through a narrow chamber and are counted using a laser, which would react with the fluorescent label, and a detector that would identify the fluorescence of those labeled cells. Advantage can easily differentiate living cells from dead cells. You also have what they call a culture counter, and these are machines that count cells in culture as they pass through a thin tube. Here's an example of a hemocytometer. Actually, I used them when I was doing tissue culture many years ago. You would take a small sample, let it flow across here on this counting chamber slide, put a cover slip on top of it, and then read it by a microscope. And you would have a certain mathematical approach so that wherever in your grid you had cells, you would count those and then multiply back up to equal how many cells are basically present in a microliter or even in a liter of cells. You could do it that way. Here's the approach for both the culture counter, which sends the cells down, but then using the flow cytometer. Because what you do is you set up the cells as being fluorescently labeled. Okay? Once they fluoresce, you have that detector, and it basically you have scattered light from all the cells, but then you're able to identify the fluorescence from the stained cells. There are certain chemicals that will tell you the difference between dead cells and living cells. Problem here, you may or may not get the same thing. There are some stains that allow the cells that are living, they will immediately eject out the dye, whereas dead cells would not. And so you can do using a hemocytometer the same approach. Now, in other ways to measure cell density, cell counts, etc., for example, using a viable plate count. This is a method to count cells based on small samples used to grow a colony. One colony from one cell. That's the way we're looking at it. And the colony forming unit, CFU. This is based on the concept of one cell will yield one colony in culture. Now, indirect counting methods. Well, The shortcoming is they can't always tell living from dead cells. Example, turbidity. Turbidity is really looking at something as it gets more cloudy. You notice that, well, if something's turbid, there's going to be a lot of particles in there. And so the greater the number of particles, the greater the turbidity. If you started off with a nice clean sample chamber and it gets turbid, you probably know there's a lot of microbes or particles in there. So turbidity, we use the density of cells based on how much light is absorbed from a light beam passed through a cell culture sample. The greater the absorbance means the greater number of cells. What you do is you take a sample, you put it into a spectrophotometer. It's a machine using light to determine optical density, the turbidity of the sample. The more density of the sample will decrease the light signal, and thereby this increases light absorbance. Now, the other way to do this is dry weight. The methods of cell count are determined as cells are centrifuged and reduced to a solid, dry pellet of cells. Another way is biochemical activity. The cell count is determined by the biochemical activity of the cell sample. Now, I'm sure you're probably thinking, well, yeah, but some of them would include in these counts, perhaps, like dry weight, dead cells. That's true. Methods for identifying microbes. Well, let's go back to what we talked about a few minutes ago, because this really begins to boil it down. First, morphology. You kind of dealt with that in chapters 1 and 2 and 2.3. Shape, size, grouping of cells. Okay, that's the beginning, but it's round. So what? Gram stain in microscopy. Is it gram positive, gram negative? Is it such that we have to use an endospore stain, an acid-fast stain, or a capsule stain? Biochemical analysis. The cell is identified by what media it can live on and by what products the cell culture makes. This is then we get into selective and differential media. Finally, the way to really be sure of something is to go right to its genome. The DNA tests, polymerase chain reaction, identification based on the genome of the microbe. Here's a concept that's called serial dilution. Let's say my initial sample is pretty turbid. I'm like, oh, wow, I can't count the number of cells in that. But if I have 10 mils here and I have 9 mils here and I take a tenth of this, 1 mil, put it into this, I now have 10 mils of broth, but I've diluted this tenfold. Mix it, do it again. I now have diluted it 100-fold. If you keep doing this for a bit, and then start taking samples out and adding them to plates, you will be able to determine how many cells are in this by how much you've diluted it and how many colonies appear. If you think about this and you go back to colony is equal to 1 cell, then you can get down to incubating and getting plates, equating back what is the dilution to what is the original sample. You can make a determination of what is not merely the number of cells here, but the number of cells here. This is a spectrophotometer. Use them, they're great machines. And basically, it's on the principle of Beer's law, which is basically the amount of light going through something, and you have the detector. If you have absolutely, you use a control, or basically a blank, as they would call it, and you have the light go all the way through, now you have 100% light transmission. Fine. You take the same, another tube, but this one is inoculated with, let's say, broth culture. You can get an idea about how much light is scattered, and therefore begin to set up a scale and also set up a plan. If you take, let's say, 1,000,000 cells and you get a certain number, you get 500,000 cells, you get up another number for percentage of light transmitted, etc. You can set up a nice arrangement to get a good estimate of the number of cells that are present in a particular sample. So, direct and indirect cell counting methods. Just go over it very briefly. Direct is microscopic count. Problem, doesn't always differentiate between living and dead cells. Coulter counter or flow cytometer, it can distinguish cells by detecting fluorescent tags and can differentiate between living and dead cells. Viable plate count, the colonies that grow from a diluted cell sample account for the original cell count. Sounds good. Okay. Indirect methods. Well, turbidity measurement using a spectrophotometer is used to measure the cloudiness of the liquid culture. Dry weight, dry mass measurement. Biochemical activity, the byproducts of cell growth are measured. Now, we've gotten to the point where we've been able to say, here we can identify the cell types. Here we can basically understand the cell density. What would these tools be used for? How about determinations for decontamination, sterilization, disinfection? And this is not just merely doing this with a sense of, well, we'll just expose it to one chemical like chlorine bleach. What about the different means to achieve decontamination, sterilization, disinfection, such as heat, ultraviolet light, different chemicals, etc.? Here we're going to discuss and review the control strategies aimed to reduce or eliminate microbial contamination. And we're not just considering a hospital setting. You can have in the food manufacture industry. You can have this in medical device testing. Let's get some terms down though first. When I talk about decontamination, it's remove or reduce the microbial population to render an object safe for handling. When I talk about sterilization, this eliminates all bacteria, viruses, and endospores. When I talk about disinfection, that's merely reduce the microbial numbers. Now, a variety of approaches. Temperature changes. We have to keep mindful of the decimal reduction time. This is known as DRT or D value, the time in minutes to reduce 90% of the population at a set temperature. Terminal death time. This is the shortest period of time that a given temperature must be held to kill all microbes in a sample. Let me give you a couple of points here. D values are big in the canning industry. Yep, everything from your canned fruits and vegetables, your canned meats, and everything else. And usually in those, they will go for D values where it's 17 times that. What I mean by that is the D value to reduce 90%. Let's say I'll say it's 5 minutes. If I want to go D2 value, I would go 5 minutes and then let's say another 5 minutes. So I've reduced 90%, then 90%, which would be about 99% of the population. So think about this. If I keep adding on the time, I have a better chance of removing more than 90%, maybe 99.9999999. Now, why would they do that in canning? Well, canning food is very important. You don't want to have stuff that's contaminated to make your clients sick and to make your customers ill or sue you. You want to have a safe food product, and that's always required. Thermal death time. Shortest period of time that a given sample, a given temperature must be held to kill all microbes in the sample. Clear example, pasteurization. When you run milk today, they can do what they call ultra high pasteurization. They run the milk through a tube that has 165 degrees temperature. And you run it through the tube for 15 seconds. It kills all the microbes. Now, there are different approaches to doing this. They don't want to lose the milk quality, but they want to make sure that they achieve the thermal death time so that none of this milk would ever go bad because it was still having some microbes still present in the sample. All right. Now, we've talked about minimum temperature need to kill all the microbes in the sample. That's the thermal death point. What about autoclaves? Well, it's interesting about autoclaves. They're a machine that applies steam with pressure, keep in mind pressure, to sterilize microbiological media and associated medical or lab equipment. There are some limits. Some plastics and some other materials will not handle the high temperatures and the high pressure. Therefore, if you're going to use materials that have plastic, etc., they use another approach to the sterilization. For example, E-beams or gamma radiation. This is particularly known for surgical disposables. Okay. But with autoclaves, you can achieve, if you hold the pressure, to bring the temperature up higher than the boiling point, which would be, of course, 212 F. You want to get it up to 240 F. This way, you achieve a higher heat to destroy the endospores that might be there. Hey, 212 might kill bacteria, but the bacteria endospores, you have to go much higher in temperature. Let's just talk about some of the convenient situations that we might deal with ourselves. Boiling. This is used to reduce the microbial numbers for drinking water, and sometimes the boiling is necessary. You may have heard where situations where they've had an earthquake or a severe disaster that's disrupted and possibly contaminated some of the water supplies. So they tell everyone, boil your water first. Okay, pasteurization, which I've already talked a little bit about. This is the application of a moderate heat to eliminate pathogens and reduce microbes to safe levels for select products. Milk, juice, other containerized fluids, okay? Soup, things like that. Ultra pasteurization is the use of ultra high temperatures at low times to kill off all the microbes. The food quality is not changed, but by ultra pasteurization, in some cases now, in some countries you'll find that this is how they process, I think it's in France and some of the other places in Europe, where they ultra pasteurize the milk to such a degree you don't even store it anymore in the dairy section, in the sense that the dairy section is not refrigerated. They're in tightly sealed packages. They've been ultra pasteurized. They will stay good until someone picks up that package, goes home, and opens it up to have a nice glass of milk. Okay? Then we have also dry heat, and this is using hot air ovens or incinerations for sterilization or disinfection. Sometimes with certain glassware that you cannot put it into an autoclave, so the way that they would do it, it takes a little bit longer in time, but you raise the temperature much higher, let's say 250 Fahrenheit, 300, and you have basically disinfected it, sterilized it, etc. Okay? And here you see an outline of an autoclave. You have a sealed door here so that the steam comes in. The inner chamber maintains a select amount of pressure. If there is any of the steam that gets converted to water, you can have that exhausted out. But what you can also do is raise this temperature to such a degree that you can get up to 240, 250 degrees Fahrenheit to destroy any endospores that might be in, let's say, culture media, things like that. Here's an example of an autoclave, very short one, and they are sterilizing surgical instruments for use, probably dental instruments, things like that. Here we have a process, and I believe that's milk, where they're going to do a rapid pasteurization or a rapid decontamination of this milk, and they run it through quick tubes, and then it heads off from there right over to the packaging. Now, as I said, there were several other ways that you could sterilize or disinfect things. One of them is high energy that can serve for disinfection or sterilization in the form of radiation. Now, ionizing radiation, which would include gamma rays and X-rays, X-rays are also referred to as E-beams, generate reactive ions that kill microorganisms and inactivate viruses. Usually this is by damaging the nucleic acids. So if you damage the genome, that's it. The party's over for those microbes. It's used for sterilization of packaged medical products, sutures, disposable um, disposable cauterizers and things like that. Okay, it's also used for pharmaceuticals and foods. Now, non-ionizing radiation, that's where you get into ultraviolet spectrum. And what UV does is it changes the bond structure of DNA. It results in mutations and ultimately in cell death. What happens is usually the DNA, if you have two thymines together, the UV light will form a covalent bond between them that forms what's called thymine dimers. Uh, and so it disrupts and eventually destroys the organism. This is used to disinfect operating room surfaces. Then we also have filtration. This is decontamination of large volumes of fluids, whether that's water or air, via microbe capturing filters. You may have heard of HEPA filters. Those are high efficiency particulate air filters. And so the particles will get caught in the filter, and yet you have large amounts of air moving through it. Here we have a means in which we're using ultraviolet light on this culture, this hood. And the purpose is that it's sort of, you could say, well, it's the end of the day or the beginning of the day, whatever you do. And what you do is you expose this to ultraviolet light for a certain set period of time, and the surfaces are all basically decontaminated, ready to go to work. HEPA filters, you can see how this has a wavy format with a filter frame. The airflow goes through it. This is a continuous sheet of filter medium. And so what you have, because of the randomization of the fibers, is you're going to capture various types of particles, dust, and bacteria. Now, in certain filters, you can have them set for a unique size. In this case, you can take the fluid here, run it through this filter, and basically have filtrated water with your, I'm going to presume that's for a vaccination or for a injectable here, and it would be suitable. Now, this is a water straw, or it's called a life straw. What happens is that the water is needed. It sounds like I was going to say the water is dirty. Yes, the water may be dirty, it may be murky, it may also contain various viruses, bacteria, uh, and even protozoal organisms, etc. But this filtration device right here filters out all the microbes, etc., while the person adds suction here to draw the water up through the filtering unit, and the water is drinkable. Now, we've also got to get into germicides. These are disinfectants, antiseptics. You've heard about them all, but we'll go over them briefly. It is important to keep this in mind when we start dealing with other forms of microbes, whether we're talking bacteria, fungi, protozoans, helminths, etc. Disinfectants are those that are used to treat inanimate objects, okay? Tabletops, doorknobs, scalpel, things like that. Antiseptics, though, are applied to living tissue. Now, there are three tiers of germicides: high, low, and intermediate. In the high level agents, these destroy all microbes, bacteria, fungi, and viruses. The intermediate level agents destroy all bacteria, viruses, and fungi, but cannot or do not destroy endospores. Low level agents destroy bacteria, but not Mycoplasma tuberculosis. They will destroy fungi and some viruses, but not endospores. Now, you want to keep in mind that provided that all of the above are at low concentrations. The point being is that if you have a very high concentration, some of them may actually survive the low and intermediate level, and even in some cases high level, if there's sufficient number of living cells that can withstand it for the short period of time. Okay? When I talk about a microbiocidal, CIDL should tell you it's killing. They kill all the microbes. Microbiostatic inhibits microbial growth, but does not necessarily kill them. Now, medical equipment that requires regular decontamination is classified into 3 tiers: critical equipment, semi-critical equipment, non-critical equipment. Critical equipment must come in contact with the sterile body sites or vascular system. These include stents, surgical tools, implants, urinary and cardiac catheters, and therefore they require high-level disinfectants. Semi-critical equipment, they come in contact with mucous membranes or non-intact skin. They must be free of bacteria, viruses, and fungi. They may allow for low level of endospores. This usually does require high level of disinfectants, but sometimes middle level is acceptable. Non-critical equipment comes in contact with the surfaces of the patient rooms, patient skin surface, skin contact devices such as stethoscopes, blood pressure cuffs, etc. Low levels of disinfectants to remove microbes is okay in this case. All right. Now, when we talk about germicides, and yes, you will definitely need to know this stuff. Alcohols are considered intermediate level. They target proteins and lipid membranes. Aldehydes, this is where you have formaldehyde and glutaraldehyde, is high to intermediate level in its capability of disinfecting, and it targets proteins and nucleic acids. Phenols, now this goes back to Lister because his first work was using carbolic acid, which is really phenol. And the derivatives to this day we use triclosan. Triclosan, by the way, you'll find it in everything, including in toothpaste in certain cases. Phenols are intermediate. They target proteins and lipid membranes, but unfortunately they can irritate skin. Halogens include chlorine and iodine. They can be high, and I'll say why they can be. They target proteins and nucleic acids, but they're limited due to the organic matter present. So let me give you an example of that one. Someone says, "Hey, look, we've got to use chlorine here, etc." But what has happened is, let's say it's pool water, and you say, "Well, chlorine will disinfect it. There's no problem." Yeah, I know, but you don't understand. This was not just a little bit of contaminant. This was an animal pooped in the water. Okay, whether we're talking a cow or whatever. So, and I'm not trying to be crude here. I want you to think about this. When I say organic matter, the larger the amount of organic matter tends to blunt out and use up or chemically react with the chlorine, and therefore its capability of disinfection has been greatly reduced. Okay? Peroxygens, including hydrogen peroxide, peracetic acid, this is very high. It targets nucleic acid and proteins. Ethylene oxide is very high. It targets proteins and nucleic acids. It used to be the standard for decontamination in hospitals for surgical equipment and things like that. The problem was it was considered a greenhouse gas, and so all hospitals had to phase out ethylene oxide. Detergents are low level. They primarily target lipid membranes. And then we get into the what you might call detergents from the sense of anionic or cationic. The anionic, for example, soap, sodium lauryl sulfate. The cationic is quaternary ammonium compounds, QACs. These are bactericidal and sporostatic. Sporostatic meaning that they will inhibit spore generation. Bactericidal, they will kill the bacteria. But you have to keep in mind that they have limitations. Now, to give you something to think about, which one of these would you think that people would use best and effectively in situations of COVID-19 contamination? Well, they're starting to, at this present date, which is May 31st, 2020, they're starting to open up places, including restaurants in some states, churches in some states, etc. And I went to church even today where they asked afterwards, would you, those that can, please stay afterwards so that we can decontaminate uh, the church. Now, some of these substances might be very good, but they would also have effects on cloth, skin, wood, varnished wood, and other things that you have to think about. So, in certain situations in a hospital surgery room, you would have certain materials that would, no question, definitely want to use it. You would have other materials that would be used only in limited means or for specific substances. Okay. Um, in other cases, you would only be able to use this in a limited approach. For example, you don't want to have anything that irritates skin. Remember, Lister would pour phenols, carbolic acids, right into open wounds in the skin. Well, that's all he had for a disinfectant at the time. Now, later on, we have other types of disinfectants that are far less irritating to the skin, just to get you to think about things. And here you can see these lists. Again, you don't have to memorize everything, but just understand some of the pros, the cons. Some of these can react with some of the substances you want to decontaminate. So it's almost as if you want to say, if it's a plastic handle on a scalpel, is it really worth it? Also, you have to think about certain situations where they might leave residue, where they might be toxic, where they might cause damage to fabrics or organic matter, or might be corrosive, all right? Or they just might be flammable. Now, many factors have to be considered to select an appropriate germicide, as I've been talking about here. And these are some of the questions that have to be considered. How is the object used? How likely is the transmission of pathogens? Must it be sterilized? Germicide reactivity. Does the germicide react, corrode materials, burn living tissue? Would treatment distort functional use? The germicide concentration and the treatment times. Is the time too long to treat between uses? Is the concentration of germicide useful for sterilization or merely just disinfection? The types of infectious agents that have to be controlled. The presence of organic or inorganic matter on the treated item, and the impact of germicide residues on equipment use. Finally, the germicide toxicity, and this includes environmental impact or the disposal of the used germicide. These factors have become paramount in some hospitals when they have to consider, we use this, how are we going to be able to effectively dispose of it? We may not be able to just pour it down the drain. Okay, and here's a couple of reminders. This is an antibacterial hand wash. You can see it has triclosan at 0.46%, very low amount. Now here is something where they're taking in and basically sterilizing this. I think this is I want to say, and it looks like it might be something like an endoscope. One of the problems that has occurred with prions, by the way, is that prions have been very, very tough. They have been reported to demand high temperatures. They may have to require also longer periods of time of exposure to glutaraldehyde. As I told you, there was a situation where they had actually a stereotaxic surgical device that basically they thought they had properly decontaminated until later on they found out they basically had prion contamination, and it was passed on to 13 other patients that had the same stereotaxic brain surgical tools used. So in all of these cases, they want to maintain sterility, and they want to maintain the optimal performance of their procedures to prevent contamination to subsequent patients. Now, there are a couple of factors here to bring up that would be helpful, so we're going to do it right now. The different control methods work for different microbes. Mycobacterium control. One of the problems you have is you have to deal with the waxy cell walls, because that is protection to many disinfectants, and therefore high levels of germicides are necessary. Since transmission is airborne, you're going to require masks, filters, HEPA filters for the air, as well as isolation. That's going to absolutely be necessary with these mycobacterium or tuberculosis patients. Endospore control. Endospores are resilient to many means, including radiation, drying, boiling, etc. This requires the use of autoclaves, bleach, hydrogen peroxide, vapor at high heat. Viral control. Now, remember we talked about in a chapter before about naked versus enveloped viruses. With enveloped viruses, which have that lipid outer layer, okay, heat, drying, or detergents will inactivate them. With naked viruses, those are the tougher ones, because really what you have is a protein capsid. You have to go to high germicides, glutaraldehyde, or a chlorine-based disinfectant there. For protozoan control, the spore phase of the protozoan can resist chlorine treatment in water. That's why you're going to be required to use ultraviolet light ozone treatment of water. For vegetative protozoans, they can be killed by boiling or filtering. As I mentioned, prion control has been very, very tough. They can withstand autoclaving and chemical sterilization. The remedy of combined chemical and increased heat and pressure during autoclaving has usually been what has been effective. And I just want you to go over these in this image. This is a good way to kind of summarize physical methods versus chemical methods of controlling microbial growth. And now we're finished. Now, I encourage you to review the visual summary on page 223, review the chapter overview on page 224. Review the end of the chapter questions on pages 226, 227. When you're working with Mastering Microbiology, your homework is due by 11:59 p.m. Friday of this week, okay? For your quiz in Mastering Microbiology, that is due by 11:59 p.m. Saturday of this week. I strongly encourage you to review the videos in Blackboard for the lecture content, because those videos are going to be very helpful to reemphasize and expand on certain components of this chapter. Our next section will be involving microbial biometabolism. I know it's a mouthful. So microbial metabolism is what we're going to be dealing with next, and that's going to be chapter 8. And I hope you have a wonderful day.